Welcome back, friends. In this lecture I am going to tell you about epistasis. Okay, I already made earlier videos regarding epistasis, but in this video I am going to compile everything and all the aspect of epistasis regarding different examples, and we'll also talk about the different ratios of the epistasis. So let's talk about it. What is epistasis in the first place? Epistasis means this is a kind of gene interaction. This is a type of gene interaction where one gene masks the effect of another gene. Okay, let's say the gene A. It is masking the effect of another gene, that is gene B. Now two genes will interplay in this epistatic events. Okay, now the gene which is masking the effect of the gene is known as epistatic gene. This is known as epistatic gene, and the gene whose functionality or the expression is masked or prevented is known as the regulated gene. Okay, this is epistatic, this is regulated, because this gene's activity is regulated by the epistatic gene. Okay, so there are different types of epistasis possible, okay, based on the type of gene that are involved with the epistasis. For example, if the epistatic gene in effect shows only in the dominant form, it will be known as dominant epistasis. If the epistatic gene acts in the recessive form, then it will called as recessive epistasis. If either dominant or recessive form of the epistatic gene ultimately mask the effect of the regulated gene or hypostatic gene, that will be known as dominant recessive epistasis. Okay, so these are the different varieties of epistasis, and in the future slides we are going to show different types of variations of epistasis and their examples, and we'll also talk about how those ratios of epistasis actually originated. Because normally if we consider the process of Mendelian inheritance, we know after the dihybrid cross the phenotypic ratio always comes like 9:3:3:1. That is the Mendelian phenotypic ratio. Now this ratio is changed, it's altered by means of epistasis. For different types of epistasis, this phenotypic ratio is altered and changed in the F2 generation. Remember, and that ratio will be very important for your exam perspective. If you are going for GATE exam, NET exam and all these exams, they ask question from this ratio. So let's look at it. First type of epistasis we want to deal with is recessive epistasis. In this case, a recessive allele one recessive allele actually block and prevent the effect of another gene. In this case the gene is B. So the gene A can be present in two format. AA, this is one format, this is another. Three different formats are possible: homozygous dominant, heterozygous and homozygous recessive. Now we call it as a recessive epistasis because if the gene is present in the recessive format, that is AA format, it is going to prevent the expression of the gene B. Okay, so let's look at it. The coat color in mice is the example of recessive epistasis, and actually it is coded with two different genes. First gene is capital C, and this capital C is if capital C is present, it will add the pigment. If small c is present, that means there will be no pigment. So if there is no pigment, no coloration, remember, and capital C is the epistatic gene, remember that. Small c is the epistatic gene, which is denoted here with the light gray color, remember that. Now the other gene is capital B, small b. If capital B is present, that means it will add more pigment, so the color will be black. If small b is present, less pigment, color will be brown. So if small c is present, so there won't be any pigment, so the color will be albino. That means it's completely white, no color at all. Okay, and this small c actually control the expression of capital B. So if small c is present, even though capital B is found, in the gene, it will not express. So the offspring will be colorless and albino, okay, no matter what condition it is. Here we go. There are three different formats. One is capital B, capital C. You know, in all these cases we have these three different formats, actually four different situations. If we have a capital B and a capital C in each of these locus, both of them are dominant. So in that case it is going to add, you know, capital B means more pigment, capital C means obviously add pigment. So the color will be black. Second scenario is small b and capital C, small c or capital C, whatever it is. In this case, as we have capital C present, that means presence of pigment, small b goes for brown color, so it is going to give us brown phenotype. These are the brown phenotype we saw. Here we don't know about the first locus, we know the second locus, that is small c. Now we know small c is a recessive epistatic gene, and if it's present in the recessive form, it is going to prevent the expression of any other gene, that is capital B. So even if two capital B is present, still the color will be albino. If one capital B, one small b, color will be albino. If small b, then also the color will be albino. It does not matter whatever combination these genes are present, homozygous dominant, recessive or heterozygous, small c is going to mask the effect of the capital B gene. It is going to provide an albino phenotype, which are these four. Okay. So what we find here based on this ratio, normally the ratio should be 9:3:3:1. Instead of that, we get a ratio of 9 black without any alteration. We get 3 brown and we get 4 white or albino. See there? That will be the ratio for recessive epistasis, 9:3:4. Let's move to the next one. The next type of epistasis is dominant epistasis. In this case, the gene that will regulate other gene will be in dominant form. So epistatic gene is, let's say, A. It is dominant over the hypostatic gene, that is B. Okay. In the early case, we saw the epistatic gene was in the recessive form. Here we are seeing that it is in the dominant form. Now the example of this effect is the squash color, the color of the squash fruits there. So let me go back a little bit. The color, as you see, different colors are possible: green, yellow, like yellow, white, then darker, and then we also have this reddish or orangish color. So let's look at the chart or pedigree, and we know in the dihybrid cross, after the cross in F1 generation, in F2 we should have a 9:3:3:1 ratio, and we put all them in the normal format. Now what happens in this case of dominant epistasis, that this dominant allele is capital W, which is epistatic in nature. Okay, it is epistatic in nature. Capital W goes for no color, that is, it will add white coloration. It is epistatic over another gene, that is G. G for yellow, green. Okay, G for green and yellow. Capital G for green, small g is for yellow. Okay, these are the two genes. The first gene is W, capital W and small w, two variations. But capital W is the dominant epistatic gene here. So if capital W is present, it is going to mask the effect of either capital G or small g. Does not matter. Okay, so let's look at here. 9 of them contains capital W in one of their locus. So it does not matter whether capital G or small g is present, they are going to add color white. So no color will be added and the fruit color will be white. 3 out of 16, they have again one capital W at a locus, and the second locus they have small g, small g. Again, as capital W is still present, it will not allow the effect of small g, so no color will be added. the fruit color will be white. Now here 3 of them contains small w in the first locus. In the second one they have a capital G. Now as they don't have any capital W, that means there won't be any epistatic effect. So the small w is a recessive form, and we know this is a dominant epistatic, means the gene will only be epistatic in its dominant form. So in the recessive form, small w, it will not behave like epistatic gene. So the later stage, that is among capital G and small g, any one of that can express. Now as capital G is present, it is dominant over small g, and capital G goes for yellow. So capital G is going to express itself, so the fruit color will be yellow. One of them, small w, small g, so they don't have capital W, so no epistasis. Small w will not be epistatic. So small g is present, small g codes for green. So what color of the fruit will be? Green. So now if you look at this pedigree, and if you look at this chart, actually you will find the ratio change. Like white will be 9 plus 3, 12, yellow will be 3:3, and green will be 1. So the ratio will be 12:3:1. That is an example of dominant epistasis. Let's move to the third type of epistatic event. This is called duplicate dominant epistasis. Why duplicate? Because this is a type of dominant epistasis while 2 genes are acting as a epistatic gene here. Okay, so let's look at the scenario. This is the example of kernel color in wheat. The kernel color in wheat is normally mediated by the presence of 2 genes, gene A and gene B, because both gene A and gene B provide a protein and produce a protein. Gene A produces enzyme A, gene B produces enzyme B. So for the expression of the color in this kernel color of the wheat, we can have either the expression of gene A or the expression of gene B. If gene A express, it can convert precursor into product. If gene B express into enzyme B, it can also convert the precursor into product. So we don't need the presence of both. We need either gene A or we need either gene B expression to ultimately give us the phenotype. Okay, that is why it's called duplicate dominant, because 2 genes can act as a dominant recessive, dominant epistasis. Now it could be either gene A or gene B, so there is an or between them. So look at the genotype again. The idea of the cross, the dihybrid cross, if you see, 9 of them will have a capital A and capital B in both of this locus. So one of them are dominant in both the locus. 9 of the offspring will be there. Now in this case, as both gene A as well as gene B are present, we know that either of this gene can convert the precursor into color. So here 2 of them are present, so obviously we are going to get color. So the colored kernels will be generated. 3 of them contains capital A in one locus and the other one small b. So capital B is not present to add the color, but capital A is present, which is sufficient to add the color. So we have a colored kernel. 3 of them contain small a in one locus, and in the other locus we have a capital B. So as we have a capital B, it is sufficient to add the color, though capital A is not present. So again colored kernel. So most of them will give us color because we only need one gene between them. And finally we have small b, which is 1 out of 16 offsprings. And in this case they don't have either A or B. So ultimately this plant, this organism actually in this case, it will not lead to any color. Okay, so colorless kernel will be generated very rarely, 1 out of 16. So the ratio will be 9 colorless. If we add the colorless, it will be 15, and if we add the colored kernels, it will be 15, and coloredless kernels will be 1. So 15:1 will be the ratio. That is the idea. of dominant duplicate dominant epistasis. As you see, 15:1 ratio. The fourth type of epistatic event that we want to talk about is duplicate recessive epistasis. We saw duplicate dominant. Now we will see, we saw duplicate dominant epistasis. Now we will see duplicate recessive epistasis. In duplicate recessive epistasis, the same scenario, but again two genes are present who can act as an epistatic gene, but in this case both the genes are required. So gene A and gene B, for example, we require both. So gene A and gene B. In case of duplicate dominant, we see that gene A or gene B, any one of that can take the effect, but here we need both of them to see the effect. So let's say there is a precursor. Again, we are talking about the example of coloration and production of anthocyanin. For example, we need to produce this chromogenic compound anthocyanin from a precursor. The first stage of this reaction is mediated by enzyme C, which is produced from gene C, which provide as an intermediate. The second part is also with enzyme P, which is encoded by gene P, that ultimately converted this intermediate into anthocyanin. So for the production of anthocyanin, we require both gene C as well as gene P. Okay, so if only gene C is present, we won't get anthocyanin. If only gene P is present or enzyme P is present, we won't get anthocyanin. So we need both of them to get this product. Now let's look at the Punnett square and the idea. Here you see 9 of them in the after the cross in F1 generation. 9 of them contains dominant allele in one of their locus, like C and P, capital C and capital P. That means we got both capital C as well as capital P genes over there, so they will express, so we can form anthocyanin. So we have anthocyanins produced and flowers will be colored. 9 of the flowers will be colored here. Now let's say 3 of the 16, there is only capital C in one locus, and the other locus we have small p, so there is no capital P. As there is no capital P, as I told you, if only C or only P is present, we won't see the production of anthocyanin. So no anthocyanin produced, no coloration. So white, the flower will be white. So white flower is 3. The third one, again 3 out of 16, small c in one locus and capital P is present. So here we have only one dominant allele, that is capital P, but capital C is absent. So again no anthocyanin will be produced. So again 3 flower will be white. And finally 1 out of 16 contains neither capital C nor capital P in any of these loci. So ultimately in this 1 out of 16, they will not have expression of any color because they don't have either C or P, none of them. So no chance of getting the color. So again they will be colorless flower or white flower. So now if we add this ratio, it will be 9:7, and that is the ratio of duplicate recessive epistasis. We have a ratio of 9:7, and we call these two genes as a complementary gene, because gene C and gene P, they work in a complementary fashion to give us a product, that is anthocyanin here. So if C is not present or P is not present, we are not going to get the color. The last one of the epistatic events, dominant recessive epistasis. Let's look at this one. In this case, the example is the production of malvidine in the plant Primula. Okay. Now in this case, one gene, like both the synthesis of the chemical is controlled by the gene K, capital K. K gene is actually regulates the expression of this malvidine production, and K is regulated by a gene called D. Okay, So what happens actually, the effect will be not shown if dominant D gene is present. So let me actually explain with the table. Here you see, if capital D is present, it is going to block the expression of malvidin production, because normally K produces malvidin. K is regulated, actually inhibited by capital D, so it is blocking the K expression. So let's say here 9 out of 16 they have a capital K and capital D in either of the locus. So as a result of that, as capital D is present in one of the locus, it is going to inhibit the expression of capital K. So though capital K should produce malvidin, but due to the presence of capital D gene, it will not be allowed to do that. So no malvidin production. So no malvidin and malvidin. Sorry. Okay, let's look at the ratio. So 9 of them no malvidin production. 3 of them contains capital K in one locus, in the other one small d small d. So as you see, capital D is not present. Small d will not be inhibiting capital K expression. So capital K can easily express, so malvidin can be produced. So here we have 3 of them producing malvidin. Now small k in one locus, in other capital D, and it could be capital D or small d other one. So as capital D is present, it is going to mask the effect of the K, but again capital K is not present also, so there is no chance of malvidin production. So 3 of them will not produce any malvidin. And finally, last one, small d, 1 out of 16 contains that. In this case, see, capital D is not present, capital K is also not present. So again, no chance of malvidin production, because though capital D is not present, so nobody is inhibiting the production, but capital K should be required to produce malvidin. The recessive form of K will not help to produce malvidin. So again, malvidin will not be produced. So no malvidin production. So what we get, the ratio will be 12:4. Sorry, actually I wrote it wrong here. So no malvidin production should go here. Sorry. Okay. So no malvidin will be produced. So let's put +1 here, and we now get the ratio of 13:3. 13 will produce no malvidin, 3 only produce malvidin, and those 3 produce malvidin. If you look, they have one of their allele section, one of the locus contain a capital form of K, another one is a recessive form. So this is the scenario when the dominant form of the epistatic gene will prevent the expression of the other gene, and the recessive form of that gene will help to produce that effect by the other gene. So that is the idea of dominant recessive epistasis, and the ratio is 13:1. See, so these are all the differences and different variations of epistasis, and epistasis is a prime example of gene-gene interaction, okay, which Gregor Mendel was unable to find out, but later, as we saw, that genes interact for their expressivity, and that's one of the beautiest example of the gene-gene interaction. Hope this tutorial helps you. If you like this video, please hit the like button, share this video with your friends, and definitely subscribe to my channel to get more videos like that. Thank you.